Most welcome my friend in our microbiology section notes. Let's start remaining things what we are studying, so the bacterial reproductions and growth cycles. Population growth of bacteria, how the bacterial population grow. It's a major themes. Population growth means orderly increasing. That means orderly 1, 2, 4, 6 like that cellular constituents after the binary fusion or any other reproductive pathway in the bacterial cells. Increase of mass not really reflect the growth. You cannot measure, you cannot equally define if your mass is increases, the number of the cell increases and number of the body growth increases. It is not equal because there is only increase in the size and weight of the cells is required only for the divisions. All actively growing cells mainly multiply the binary fissions. This is one of the major step in the bacteria. In binary fission, one specific cells undergo divisions to give rise to the formation of two cells. These divisions are two chromosomal divisions and cell divisions. That is means C phase and D phase. So we need to understand already. Last video we read that all these things, what is the C phase, what is the D phases. So any cell who really want to go for the division need enormous amount of the nutrients and proper cell cycles, so replication process of DNA. Let's the population growth study here. This is the in bacteria geometrically study. Geometrically population of growth in bacteria is going either 1 is go to 2, 2 to go to power 2, 2 to power 3, 2 to power 4, 2 to power 4. All these are the number of generation. If you are thinking a third generation, how much number of cells? So you 1, 2 is equal to 2 is equal to 2, 3 times you can multiply, then you understand number of cells. Just this is the formula. Assuming there is no cell death each successive generation, in this condition, one of the major condition, we are not assuming any cell is going to die, so that we can predict the number of cells in that generations. Cell give rise the double of its population. This total number of population is n in the end of the each time period. Expression is this: n is equal to 1 is in multiply 2 to power n. If there is no any cell was die, is die, will die in successive generation, the end of generation, you can calculate by this formula the number of the cells in the bacterial multiplication after the binary fission. But under practical condition, but it is just assuming condition, but in practical condition it is difficult to inoculate by ones. So this formula n is equal to n0 multiply 2 to power n. N0 is the number of bacteria at the time of 0. When there is no replication, no binary fission is going on, that time is N0, that is one cell is present into 2 to power n. That generation is a third generation, so you can multiply like this, fourth generation like this, and n is the end number of the cells. So this formula is very important for calculation of the bacterial growth cells. Now we are taking logarithm in both sides in this formula, n0 and 2. So this is coming log n0 plus that power is coming here. So you can, this power is come when you are taking log n, then is come n before that. This is log, this is n. So n is equal to this, we are going to, this is separation, this we can make the formula by mathematics. N is equal to log n minus log n0, but under log 2. Log 2, you can put the value of log 2 here, 3.301. Then you can find n is equal to 3.3 log n minus log n0. If we know the initial population, this is the initial population, then the population after the growth we can calculate by this formula. So this is the one of the population growth in the bacteria. You can calculate by the each generation under how many number of cells are present. By this is one of the good examples. You can calculate bacterial growth and bacterial cell cycles in the successive division of the cells. Hope you are liking these videos. In these days we are making the microbiology notes on the online sections. and we are coming back again with new video. Thank you.